Enzyme action and enzyme properties. Enzymes manipulate the substrate in the active site, and it takes part in the small part of the enzyme called the active site, while the rest of the protein acts as scaffolding. And the substrate molecule binds to the active site and the product is released. There's two models for the action of active sites, which are the lock and key and the induced fit. The lock and key model states that the enzyme's active site is complementary to the substrate molecule, and the active site is the lock and the substrate is the key fitting into the lock. The shape and properties of the active site are given by the amino acids surrounding it, and the amino acids form hydrogen and ionic bonds with the substrate molecule, so the active site binds to one substrate only. The induced fit model states that the enzyme is flexible and the active site isn't complementary to the substrate, and so the active site changes to fit the substrate, and the change distorts the substrate in the active site, making it more likely to change into the product. And the enzyme can change the active site's conditions like the pH and the water concentration so that the reaction is more likely to happen. Enzymes lower activation energy of reactions because I keep saying the wrong thing, it's making me sound. Right, okay. And
You're reading.
I'm not reading, I'm just looking at my notes and then I'm saying it.
You're not meant to be. It's meant to be you talking.
I'm talking.
Talking loudly at the screen.
I am talking loudly at the screen. And the enzyme lowers the activation energy because the substrate has to overcome an energy barrier called the activation energy. And then the larger the activation energy, the slower the reaction will be. But there are also inhibitors to enzymes which inhibit the activity, reducing the rate of reaction. Like competitive inhibitors have the same structure as the substrate molecule, but they fit into the enzyme active site. And non-competitive inhibitors, they don't fit into the active site, they go into a binding site at the back of the enzyme and change the active site so the substrate can't fit. 235.
So starch is the most abundant thing in the body, so we need to break it down, and this is done by mechanics in our body. The first thing is the mast salivary gland. It produces amylase, and this amylase basically is really weak, so it breaks down very little, and it goes down into the stomach, and the stomach has pancreatic amylase, which digests most of it, but the ones that aren't digested are the disaccharides, and it leaves disaccharides and it goes into the ileum, and in the ileum there are enzymes. So for maltose you'd use maltase to break it down into glucose, and for sucrose you'd use sucrase to break it down into glucose and fructose, and for lactose you'd use lactase to break it down into glucose and galactose. This is done by hydrolysis, and to put it back into disaccharides you use condensation. There's two tests that you can use, and that's the Benedict's reagent for non-reducing or reducing. Reducing is to find monosaccharides, and what you do is you have a sample and you add Benedict's reagent to it and you heat it up, and if it goes from a dark blue to a brick red to yellow, that means that a monosaccharide is present. If you have a non-reducing sugar, so that's a disaccharide, you do the Benedict's reagent and it won't react at first. So what you do is you add hydrochloric acid to break the glycosidic bonds between the disaccharide, and then you add sodium carbonate to neutralize it, and then you perform the Benedict's test again, and it should turn from blue to red. And this is proof that it's a disaccharide because it's broken the glycosidic bonds to form two monosaccharides.
Digestive system. So the first part of the digestive system is the esophagus, and this is a muscular tube which connects the mouth to the stomach, and it uses peristalsis, which longitudinal muscles to move the bolus of food down into the stomach. And it also, the glandular things lubricate the esophagus, so the bolus of food can slide down. It goes into the stomach. It's a muscular bag. It uses chemical and mechanical digestion to digest the food. The mechanical digestion, it just contracts, so it it squishes like the food up, digests it, and then the chemical digestion is using enzymes and hydrochloric acid to digest it. Then it goes to the small intestines, which contract to digest the food. And then the large intestine, the first part is the duodenum, and in the large intestine it removes all the water from the food to make it a solid, and then it goes to the rectum, which just performs digestion. The pancreas secretes enzymes to the stomach, which in some are maltase, lipase, carbohydrates. Yeah.
Right, I'm going to talk about cholera. Cholera is a disease that's caused by Vibrio cholerae, which is a bacterium, and it's a single flagellum. If you catch this disease, there's a 75% death rate. The symptoms of cholera are diarrhoea, dehydration, nausea and tiredness, and you also get bad abdominal cramps. It causes diarrhoea because the bacterium sticks to the epithelium and releases a toxin called CT toxin. The toxin then enters the epithelial cells and the chloride ions diffuse out of the cell and go into the lumen. This lowers the water potential in the lumen, and water is lost from the cells to the lumen via osmosis. Treatment for cholera is the oral rehydration solution, which raises ethical issues because a lot of children use it, but obviously they can't give their consent, so it's down to the parents to decide whether they can have it or not. The oral rehydration solution is made up of glucose, salt and sometimes other ions as well, including potassium. It uses the sodium glucose transporter protein, and basically the sodium and the glucose bind to the transporter protein, and it carries the sodium and the glucose into the cell down the concentration gradient. This lowers the water potential inside the epithelial cells, and the water diffuses from the lumen into epithelial by osmosis, which rehydrates the cells.
When you finish, press stop.
Diffusion is the movement of substances from a high concentration to a low concentration. It moves down the concentration gradient and requires no energy. For example, this is expiration, when the oxygen molecules diffuse from a high concentration in the alveoli to the capillaries, and this is the opposite for CO2, which moves from the capillaries to the alveoli. This can be used in fixed law. For example, concentration times by surface area all over thickness of cell. When there's a high concentration of substances and a high surface area and a thin pathway of diffusion, then that maximizes the diffusion rate. Facilitate diffusion is the movement of specific cells down the concentration gradient. For example, in sodium and glucose, they will move down the protein channels, carry proteins. These are in the phospholipid bilayer, which is shown in the fluid mosaic model.
The heart is a double pump, so it goes from, you've got a left and a right side, and that pumps the blood around the body or to the lungs. And then each side has its own set of veins in ventricles. There's the left ventricle, right ventricle, pulmonary artery, pulmonary vein, and vena cava and the aorta. As the blood comes into the atriums, the pressure will increase, and so will the volume, as there's loads of fluid going in. As it then travels into the ventricle, the pressure will lower in the atrium and increase in the ventricle, and so will the volume again. And then it gets pushed up and out into the body or the lungs, depending which side. That's done by electrical impulses which travel. So the electrical impulses go from the SAN node and then they travel, and that pushes it across the two atriums, and then it goes to the AVN node where there's a slight pause. Like if you listen to your heartbeat, you can hear the pause in between the beats, and then it travels in the septum. There's a bundle of his which travels down, and it reaches the bottom of the heart, and then that pushes it back up, and then that continues on a cycle, which is the cardiac cycle, and you can hear it. If you listen to your heartbeat, and if you look on an ECG, the bit where it goes like that, it's a rate. And then, so that continues. That's it.
The plasma membrane, or the cell surface membrane, surrounds the outside of the cell and is a thin, flexible layer made up of phospholipids and proteins. This controls the entry and exit of cells with the material inside the cell. Microvilli are finger-like projections which are found in cells such as epithelial cell. These increase the surface area of the cell. The nucleus is the largest organelle and is made up of a double membrane which is surrounded with nuclear pores, which controls the exit of the substances. Interior of the nucleus is called the nucleoplasm, which is full of chromatin, which gives the cell its DNA protein complex. The mitochondria, all aerobic respiration reactions take place inside this. They release energy from carbohydrates, lipids, and other energy rich molecules. Lysosomes are small membrane bound vesicles which are used to break down unwanted cells, chemicals, toxins, and organelles. Ribosomes are sites of protein synthesis and are used to make proteins for the own use of the cell, and are attached to the rough endoplasmic reticulum. Endoplasmic reticulum, there are two of them. There's a rough endoplasmic reticulum which is covered in ribosomes and that synthesizes proteins. The smooth endoplasmic reticulum is used to process mainly lipids, which has smooth surface inside. The Golgi apparatus transports proteins from the rough endoplasmic reticulum to the cell membrane. When they fuse with the small vesicles, they release the content by cytosis. Exocytosis. Magnification is basically magnifying a sample so it appears bigger, and resolution is where resolution is basically the ability to distinguish cells. Resolution is basically the ability to distinguish between 2 points of an image. Transmission electron microscopes are basically where a beam of electrons pass through a thin layer of a specimen and has high resolution. Scanning electron microscope is where a beam of electrons pass over the surface of the specimen, and electrons are reflected from the surface of the specimen, and this has a high magnification but however has a lower resolution. Ultracentrifugation is where the cell spins at high speed, and cell fractionation is where the heavier or larger cells are released first, where right at the end where the smaller cells are left.